Hello, this is Tommy. Welcome back to Chatomics. I have over 12 years experience in computational biology and bioinformatics, and I converted myself from a wet biologist to a computational biologist. So today I'm going to show you how to convert ensemble IDs to gene symbols for RNA sequencing and count matrix. So in this case, you have a count matrix with row names are ensemble IDs and columns are samples, and you want to change the ensemble ID to gene symbol. So I will show you how to do that practically in R, and there are some small places that you need to pay attention to, so make sure you stick to the end. And to do this, we are going to use an example data set from TCGA using the recount3 package. Okay, so recount3 actually curated all this RNA sequencing count metrics from TCGA and the many GO data sets. Okay, so if you don't have that package, you have to install it from Bioconductor using this command. I have already installed it, so I will just load the library and some other helping libraries. And you can run the first command, available projects, and you will see for example, it actually has the GTEx data and also the SRA data and also TCGA data. So you can filter that table by using this subset commands and only filter the TCGA data. And let's look at it. So this is the TCGA data information. So you have different project, different cancer types here. And also this is number of samples for each cancer type, okay? And let's just use one of the data sets from ACC. So essentially that's the first row of that data frame. And then you can use this create underscore RSE command and use this TCGA info, this one row data frame as the argument, and create this ranged summarized experiment object. Okay, let's do that. Okay, this is actually 79 samples, okay, from ACC. And if you just look at this, it will tell you it is a ranged summarized experiment, okay? And let's take the raw counts from this object. So you can use the @ assays and @ data and the $ raw counts. And this is the raw count matrix. Okay, with the rows are ensemble IDs, columns are sample IDs. In this case, those are UUIDs. Okay, and because it's a range summarized experiment, it actually also contains information for each gene. For example, if you look at, use the @ to access the row ranges, and you'll see actually it will show you this G-range objects. So it has the ensemble ID and also the gene symbol here, gene name here. So you can kind of already get the ensemble ID mapping and the gene mapping from here, but today I'm going to just show you how to map the ensemble ID and gene symbol from scratch. So because not every time you have this range summarized experiments available, right? Okay, let's do that, and we will need to use two package annotation DBI and this database, Homo sapiens database here. Okay, let's install them. I've already installed them, so I will just load the library. Okay, so let's just save this raw counts data matrix into a new variable called mat. So if you look at the dimension of the matrix, so you have 63,000 rows, ensemble IDs, and 79 columns and samples, essentially. Okay, so if you look at the first 5 rows and 5 columns again, So this is something it looks like. So you can save the row names, those are ensemble IDs, to a new variable called ensemble ID. So do that. So now we can actually match those ensemble IDs to gene symbols using the annotation DBI package. So you can specify the select command. This is the database we're going to use, and these are the keys, which are those ensemble IDs. And essentially, this is what we want to actually map them to, this gene symbol. And the key type will be the ensemble, essentially the ensemble IDs. So in case you have multiple mappings, you use first. Okay, let's do that. Oh, there's something wrong. Okay, none of the keys entered are valid keys of ensemble. So if you got arrows, don't panic. Read the error message and analyze what's wrong here, right? So it turns out if you look at the ensemble IDs carefully, so you see in the last digits here, you have those like dot some numbers like in the end. So those tends to be the version numbers, and for you to map the ensemble IDs to symbols, and you have to actually remove the version number in the very last. Okay, so what you need to do, okay, this is the ensemble ID original one, so you use this stir replace function from string R and to replace this dot and this number, and use regular expression here, so slash slash to escape that dot. So you want to literally match that dot, and some numbers followed, and it could be one number or more numbers, because sometimes you have like 10, dot 10, dot 12, something like that. So you want to replace them to nothing. Okay, so if you do that, oh, hold a second, I think there's some error here. Ensemble IDs, so there's a typo here. Okay, so if you do this and look at the ensemble ID again, now you remove the last digits, okay? And also, because we want to convert the original ensemble ID in the row names of the original matrix to gene symbols, we actually want to change that row names to the new ensemble ID, which we actually remove the last digit. Okay, now if you look at it again, now all the dots are removed. So now let's rerun the annotation DBI select command again. This is the database, the same commands. Then this new ensemble ID, we just cleaned up, removed the last digit, and let's map it. Okay, so it'll also show you some message. Okay, returns a one-to-many mapping between keys and the columns. Right, so let's actually look at the data. So it will return actually a data frame with two columns, the ensemble ID and symbols. The first thing you probably notice, there's some NA, so there's some ensemble IDs cannot be matched to any symbols. So that's actually there's some other places you want to pay attention to. Sometimes you have multiple ensemble IDs mapped to the same gene symbol, or you have multiple symbols mapped to the same ensemble IDs. So to actually verify that, you can use this function from janitor, get dupes here. So if say so gene symbols, that's the data frame, and if you look at how many, for example, you see there are many HNAs, for example, you have 25,000 something like that NAs, and but if we just filter out those NAs, and actually you still see many duplicates, right, for the symbols. Okay, all those are the same gene symbol, but they are mapped to different ensemble IDs, and you have 35 such for this, I think, believe it's a non-coding RNA. And but you also have other, for example, those KIR genes, and okay, those are all KIR genes. Okay. So what we can do is actually we need to clean up that data frame to remove only, to keep only the distinct gene symbols and those distinct ensemble IDs. So to do that, we can first filter those NAs and then distinct symbol, keep all equals true, and also distinct ensemble. Okay, so let's do that. And now if you look at the dimension, now you only have 35,000 rows. If you look at the original data matrix, it is like 63,000, something like that. Okay. You can actually just verify them, okay? After you keep only the unique ensemble ID or gene symbol in the data frame, and you can try it again. Now you don't have any duplicates. Same thing for here. Okay, now we need to actually subset the original data matrix first to, for example, this is the data frame unique, and then this is the ensemble ID column. So if we do that, so those are the ensemble IDs, and then you can supply that vector and then subset the original matrix. If you do that, and if you look at the dimension subset, you only have 35,000 rows left. And if you look at the first several rows, and then also look at the several rows of that gene symbol data frame, they are actually in the same order. So what I'm telling you is that the first row here will be the first row here, okay? Because you use this column to subset the original matrix. So the matrix will be reordered. The rows will be reordered as the column in the original data frame here, okay? And this is actually important. I will show you why, okay? Then because the order is the same, right? So we can actually verify that. So the row names of that subsetted matrix and the gene symbol, essentially this data frame and this column, and let's see whether they're the same. So use the or.equal function and see they are the same. So equals true. So now you can comfortably or confidently replace the original row names of that subset matrix with the column, with the symbol column, because now they're in the same order, right? So you can just use this column to replace the original row names. So now if you do that and look at the subsetted and also remapped gene ID matrix, it will be gene symbol versus the sample IDs. Okay, so I hope this video showed you actually how to actually map different gene IDs, and also there's some things that you need to pay attention to have multiple ensemble IDs mapped to the same gene symbol, and vice versa. So you have to pay attention, and you have to subset the original matrix, right? Well, the other way is also if you want to, if there is, okay, no gene symbol can be mapped to the ensemble ID, you can also keep them, the original ensemble ID, and only convert the ensemble ID that can be mapped to gene symbol. But I tend to actually just remove them, and if there's no gene symbol that it can be mapped, probably those are not really important genes. That's just in my own opinion. Okay, that's it for today. Happy learning. Subscribe. Make sure you subscribe to ChatRNaseq, and the script for the code, you can find it in the description of this video. See you next time.